The methods needed to prepare your samples to be read in the Arbor Assays DetectX kits would depend on the type of samples you have. The most important thing to remember is that you will obtain the best analytical result if your samples are in the identical buffer or matrix that the kit standards are made in. So dilution or redissolving a dried sample in the assay buffer supplied with the kit is very important. Sample preparation methods will depend largely on the types of samples you have. Solid samples, such as fecal samples, will need to be extracted in either ethanol or methanol. The extracted sample will be centrifuged to remove debris, and the supernatant may have to be either diluted in the supplied kit reagents or dried down in a vacuum centrifuge. Some kits allow serum or plasma samples to be diluted in the dissociation reagents supplied with each kit. Please note, only use the dissociation reagent that comes with the kit you are using, as the formulation of the dissociation reagent is optimized for each kit. Kits that do not contain dissociation reagent may require an organic solvent extraction. Please go to www.arborassays.com for protocols for extraction for most samples. For cell or tissue samples, you may have to homogenize the sample. We would recommend searching YouTube for a suitable video for your type of sample. For urine samples, simple dilution in the kit, buffer or diluent should work for your samples. You may have to go to PubMed to find typical urinary concentrations for your molecule. If your samples are a single time point, you should reference the concentration of the molecule to the urinary creatinine level in your sample. The use of urinary creatinine allows you to reference your sample antigen level to a marker that takes into account the excreted volume of urine. Arbor Assays DetectX urinary creatinine detection kits are referenced to the National Institute of Science and Technology creatinine reference standard. Saliva samples for most analytes should be collected using a cotton device such as a Starset Salivette. If you are not using this type of saliva collection device, collect the sample in a plastic conical tube and freeze at -20 degrees Celsius. Thaw and centrifuge the sample and remove the clear supernatant. In most cases, the supernatant can be diluted in the kit assay buffer prior to analysis. Special sample preparation instructions. When analyzing cyclic nucleotides such as cyclic AMP or cyclic GMP in the Arbor Assays DetectX kits, it is important to use the sample diluent supplied in the kit. To prepare the sample, a phosphodiesterase inhibitor such as IBMX should be added to the sample. We supply IBMX as catalog number P019 in two different sizes, a 100 milligram size and a 1 gram size. Most samples using media can be read in our kits using standards prepared in the identical media. All small peptide samples, such as oxytocin and argvasopressin, must be extracted using the extraction solution provided with the DetectX kit, or by using one of the recommended sample extraction methods in the kit protocol. Non-extracted samples will give rise to grossly elevated concentrations of these types of peptides.